Welcome to BioMindscape. Today we are going to know about cell cycle. The cell cycle is a series of events that a cell undergoes through growth and division, resulting in two identical daughter cells. This process involves two major phases: interphase, where the cell grows and prepare for division, and mitosis, the actual cell division. Interphase is further divided into G1, S, G2 phase. So interphase is a period between cell division where the cell growth, copy DNA and prepare for mitosis. So G1 phase, the cell growth physically and increase the volume of both protein and organelle. S phase, the DNA replication occur, creating two identical copies of each chromosome. G2 phase, further growth and preparation of mitosis, ensuring all necessary material are available. M phase, mitosis phase. This phase is when the cell actually divides, resulting into two daughter cells with identical genetic material. Mitosis is a process of dividing the nucleus and its content, ensuring each daughter cell receive a complete set of chromosomes. So cytokinin, the division of cytoplasm, separate the two newly formed nuclei into distinct daughter cells. So in essence, the cell cycle ensures accurate DNA replication and segregation into two genetically identical daughter cells, which is essential for growth, repair, reproduction in multicellular organisms. G0 phase, also known as the quiescent phase, it is a stage of cell cycle where cells are more metabolically active but not actively dividing. G0 phase allows the cell to pause their division and enter a resting state until they are needed. It's also important for tissue repair when cell in G0 can be called upon to divide to replicate damaged cell. The interphase is made up of G1 phase, S phase where synthesis takes place, and G2 phase, that is the gap 2 phase. And M phase consists the mitosis where prophase, metaphase, anaphase and telophase happens. So prophase where the chromosome condense and become visible, the nuclear envelope break down and the mitotic spindle began to form. Metaphase where the chromosome align at this metaphase plate attached to the spindle fiber. In anaphase, the sister chromatids separate and are pulled to the opposite pole of the cell. And in telophase, the chromosome arrive at a pole. The nuclear envelope reform around each set of chromosome and cytokinins begin. Now cytokinins is what cytoplasm divide, resulting into two daughter cells. There are various cell cycle checkpoints. So what is this checkpoint? G1 checkpoint checks the DNA from damage and cell size before entering the S phase. G2 checkpoint ensure the DNA replication is complete and DNA is undamaged before entering the mitosis. Spindle checkpoint monitors the correct attachment of chromosome to the mitotic spindle before anaphase. These are the most important cell cycle checkpoints: G1, G2 and spindle. Cell cycle checkpoints. They are the regulatory mechanism that ensure the proper execution of each step of cell cycle. It checks, it monitors if any error is there or not, the cell cycle events are working properly. It ensures that cell division must be accurate and can induce the cell death. So let's know the checkpoint. This G1 checkpoint, G2 checkpoint, M checkpoint, that is a spindle checkpoint. The checkpoints are important for maintaining the genomic stability. It checks whether there is any damage or incomplete chromosome to the next generation, and it prevents cancer. If any defective or inactive checkpoints is there, if any defect is there, so it can remove it. So the checkpoint can contribute to genomic instability and increase the risk of cancer. Theoretical potential. The targeted cell cycle checkpoint may offer new strategies for cancer treatment, as these checkpoints can be exploited to kill cancer cells. G1 checkpoint. The checkpoint occurs at the G1S transition and checks for the cell size, protein reserves and DNA damage before the cell enter the DNA replication. G2 checkpoint. It is located at the G2M transition. This checkpoint verifies the DNA replication is complete and there is no DNA damage before cell entering the mitosis. M checkpoint. This checkpoint occurs during the metaphase of mitosis and ensure that all the chromosomes are properly attached to the spindle microtubules before the cell proceeds to anaphase. Pause the video and note down the important points